Okay, good morning, everybody. I'm Catherine Chauvel. I'm the president of the European Association of Geochemistry. Today, it's my great pleasure to start this session and this day with the presentation of the 2023 EAG Urey Medal Lecture. Harold Urey was a physical chemist whose pioneering work on isotopes earned him the Nobel Prize in Chemistry in 1934. I'm not sure all of you know that, neither did I before. And later, it led him to theory on planetary evolution. Harold Urey was a true pioneer, and the EAG has named its highest honor after him in recognition of outstanding contributions made to geochemistry over an entire career. Our 2023 Urey medalist is equally such a pioneer, so it's a great honor to present this year's Urey Award to Bo Baker Jørgensen. Please come. So in case you were at the plenary on Monday, Bo received his medal at the plenary. And so now I would like to invite Antje Butchus, I'm not sure how to say your name, to tell us more about Bo and why he received such prestigious awards. Thanks a lot.
So good morning, everyone. And of course, it's a great pleasure to introduce you for a short moment in time to Bo, the recipient of the Harold Urey Award. And it's fantastic. It's just, I have a little bit of goosebumps thinking about the connection. And of course, the connection is simple. It is about dedicating your entire oeuvre to linking chemistry to life. That is so important. We are still beginners in so many ways when you think about that. This planet, the only one we know that has life, that life makes the chemistry of that planet, but also the planet totally changes the way in all of its chemistry that life can thrive. And we are in the middle of the negotiation of a future that is unknown to many. And I find it fantastic to link the two, Bo and Harold Urey. Of course, when you think about it, you might wonder, and so the Miller-Urey experiments are so fantastic. Students love it if you teach the evolution of life and you go into that experiment. But I always wondered, like, why didn't they do more with sulfur? And I'm sure that Bo, had he written a citation to Harold Urey, would have written some critical notes. Experiment about the origin of life should be with sulfur, shouldn't it? Especially when you claim you look at volcanic gases. But there is this fantastic paper about that they really did a lot of sulfur experiments, just that Stanley Miller never published them. In 2007, after his death, vials were found with all the sulfur experiments. Please look it up again. It's so cool to tell that story of forgotten samples. And when they did that, they found that, of course, many more amino acids are there when you include sulfide in the original Miller-Urey experiment. And so today we cannot think Earth and life's emergence without sulfur. And Bo, in his achievements, and you can read all about this also in his PNAS profile, he developed a way to approach sulfurous life. He did it with microsensors and macrosensors, as you can see from these high-tech images of his various laboratory works. And he developed a keen, big love for the field work, which we all share, don't we? So I'm standing here, of course, only on behalf of the true nominators. That's Don Canfield, Fumio Inagaki, and Ken Nielsen. And when you think about that, of course, these are our heroes, amongst many others, some of which are in the room, for trying so hard to make us understand this diversity of life and the ways that life can tap into chemical resources and change earth and element cycles, and we just know a tiny little bit of that. The estimate still is that of all of the microbes, then can do so many more than we eukaryotes, that we still have not even captured 10%, and forget about cultivating them. That's a tiny fraction of what is out there. So the citation is, his specialty is biogeochemistry, geobiology, and microbial ecology, fundamental developments to monitoring the chemical nature and rates of microbial processes in nature, while developing unique modeling approaches to unravel the relationship between microbes and the chemical environment. And this I find really critical and would fully support, that you can go to these crazy environments that Earth holds for geochemists. And of course, you want to discover, you see all of the strangeness of these environments. You have your technologies, your instruments, your methods with you, but it also depends on yourself going there and getting a feeling for what it is. Stanley Miller said, when he was asked why he did so little with sulfur, he said, it smells foul, I get sick, and I have to puke if I last longer than 2 minutes with something that is sulfur. And luckily for Bo, it's different. He totally loves sulfur, and he can sniff it from very far away. And I was witness of many fantastic, foul, smelly, bad sediments where Bo still was in with everything he has. So I would like to end this very brief citation. I could go on for hours telling the real research content, but this is just to intrigue you, and we now get a full lecture on sulfur metabolism. When I read through the nomination package of the three, all three I cherish very much, I found one citation bit very, very important for us. It says, one cannot overstate Bo's ability to inspire his colleagues and students. Through his long career, Bo has educated many of the current leaders in microbial ecology and biogeochemistry while introducing microbes into biogeochemical thinking, an idea that permeates the emerging field of geobiology. We geobiologists, of course, think we are no longer emerging, we are fully there. And the rest that thinks differently will have to learn. But I also see in the citation this critical thing of being together, teaching each other, listening to each other, coming together. And so I love this fantastic, curious shot of Bo working so hard and his colleagues who love him very much, putting a sign on the box he's sitting on. That's a scientist, do not feed. Thank you, Bo, for all your hard work. We love you.
Affect your entire career. I started developing an S35 radio tracer technique to measure the rate of sulfate reduction in marine sediments. This is what I've been doing on and off ever since then. Since that start, sulfur became my favorite element and marine sediments my favorite environment. My first PhD student was Niels Peter Rebsbæk. We challenged him to build oxygen microelectrodes to make the first microscale measurements of oxygen in marine sediments, which he did. This became a true breakthrough in microbial ecology and was followed up by Niels Peter with a range of novel microsensors and exciting results. Today, he and I share an office and he's about to retire. Sad. It was also a great inspiration when Don Canfield came to work with me as a postdoc a few years after he finished his PhD with Bob Berner. He was a part of our pioneer group when we founded the Max Planck Institute for Marine Microbiology in Bremen, and he established isotope geochemistry in our department as a new tool to understand the dynamics of the sulfur cycle. And I owe a lot of my understanding of sulfur geochemistry to Don. Equally inspiring was Antje Boetius, who came to our Max Planck Institute in 1999. And within a year discovered the consortia of archaea and sulfate-reducing bacteria that oxidize methane anaerobically. This has been one of the greatest breakthroughs in the study of anaerobic methane oxidation, and Antje's fluorescence micrographs of these consortia have become iconic for the process. Throughout my research, it's been a primary aim to understand both the biogeochemical processes as they occur in the environment and the organisms responsible for them. I have relied strongly on experiments to measure the rate of microbial processes such as sulfate reduction. This has the strength that individual processes are directly detected even when they are concealed by cryptic cycling. It has the weakness that each experiment is a manipulation of living organisms that may alter their metabolic rate if they're not treated right. We still have some work to do to reconcile process rates calculated from transport reaction modeling and from experiments, and I'll talk about this shortly. The Urey Award is given for contributions that advance geochemistry over a career. When I see what the young scientists in our field are doing today and what they're discovering, I continue to be impressed and excited. I wish I could start all over, knowing what we know today. It may be difficult for me to admit my age, but the signs are clear. My grandchildren are growing up and my PhD students are starting to retire. But I'm still hired at Aarhus University, and it's still difficult to start doing science. After many years of administration, I'm painfully aware that I've gotten rusty in the lab and that it's the young people who use the new methods and instruments and who know how to do it. But I at least still know why we do it. So again, Antje, thank you very much for your kind words, and thank you to the European Association of Geochemistry and to Catherine Chauvel for the Urey Award. Thank you. Yeah, thank you. Yeah, so the story I will tell you today has to be a small part. Is there an echo here?
Yeah, so there's somebody with a...
Okay. I can only give you a very limited story about the sulfur cycle, and I decided to limit this to sulfate and sulfate reduction in the seabed. And you may say, we know a lot about that. You can read about it in the textbooks, so what's new? I think there's still surprises, and I think there's really serious work for us to do still, and I'll show some examples of that. So we're going out to sea. Here's an example where we go out, take gravity cores, do high resolution sampling for pore water chemistry, solid phase chemistry, process rates, microbiology, and what I will talk about today is not really about the microbiology, but about the processes. So what I will talk about are what controls sulfate reduction in the seabed, and how do we measure these rates. And I'll try to give you an impression of the difference between what we learned from modeling and from using sulfur-35 radiotracer, and what is the difference between net rates and gross rates of sulfate reduction. Then I'll talk briefly about substrate kinetics, temperature control, and then discuss how can we best determine the sulfate reduction rate in the global seabed. So when we go out, take sediment cores, they may look like this. You have an oxidized surface layer, and in this sediment there's sulfate reduction all the way, also up in the oxidized layer. This is in fact where we find the highest rates of sulfate reduction. And we have been able, for the first time a few years ago, to measure both the rate of sulfate reduction and the rate of sulfide oxidation using radiotracer. And what that shows is that, well, highest rates in the surface, but there's 100% recycling of the sulfide that is produced, which means that the gross rate of sulfate reduction is very high. The net rate of sulfate reduction is 0. And this is the extreme case of the sediment surface. Then they start to come together as we go down into the sediment. How they come together is really not well defined. This is one of the very open questions in sulfur geochemistry. People model sulfate, they don't model sulfide. So when we then look at the entire sulfate penetration, I'll be talking about the sulfatic zone where there's sulfate, the methanic zone where there's methane, and then the sulfate-methane transition. And we can measure sulfate reduction down through all these depths. And here in the middle is an example where the dots are individual measurements of sulfate reduction rate, the highest density that we have ever achieved. And from this, you can then make a log-log plot and a linear correlation that shows that the rates of sulfate reduction are decreasing by a power law with depth and with age in the sediment. And a power law is very non-intuitive, and it's often not understood. And if you look at the graph here, between the sediment surface and 3 meter depth, the sulfate reduction rate drops 10,000-fold. You would say nothing is happening in the deeper part. 85% of the entire sulfate reduction rate takes place in the top 30 centimeter, which is where you have bioirrigation, which means that you don't even really see it in the sulfate gradient. And when you look at the graph, it looks as if methane is really guarding the depth of sulfate penetration. But when you compare the methane flux and the sulfate reduction accumulated, only 2% of the entire sulfate reduction is fed by methane. So it's very counterintuitive. Are the rates right? Well, there's one way to check them. We have shown that the rate of mineralization is independent of whether the terminal process is sulfate reduction or methanogenesis. So we can extrapolate this trend down through the entire methane zone, and from that calculate the total methane production and compare it to the diffusive flux of methane coming back up, and they match quite well, which indicates we have the right order of magnitude of these rate measurements. So with depth, sulfate reduction is taken over by methanogenesis. And as I said, the rate of mineralization is really determined by the initial steps of degradation and not by the terminal. The sulfate reducers, the methanogens, are keeping their substrates at the energetically lowest level that they can. They're taking it up as fast as they can. So we have shown in a number of experiments how is the affinity of sulfate reducers to sulfate. How effectively do they take up sulfate? And I have done such experiments. Others have done it in the 80s. And you see here 2 examples of how the rate of sulfate reduction depends on the concentration of sulfate up to full seawater, 25 millimolar here on the scale. And you see that at lower concentrations, the rates drop with half level at 1.2 or 1.6 millimolar, the apparent Km for the process in a Michaelis-Menten plot. This is not a very high affinity. I could never understand these curves. And one of the reasons I couldn't understand it is that if we compare this with the affinity of the sulfate reducers for their substrates, they don't match. So here are the substrates. These are measurements by Clemens Glombitza of the main volatile fatty acids, formate, acetate, propionate, butyrate. And you see that, well, highest concentrations are of acetate, around 10 micromolar. Of the other is a micromolar or a few tenths of a micromolar. And when you then look at the turnover of the substrates, they differ tremendously. If you look at the acetate, it has a turnover time of about 1 hour up at the sediment surface and 1 year at depth at 100 meters. I should just say the depth scale here goes from 1 millimeter to 100 meters. So it's the entire sediment column, and it includes both the sulfatic zone, these are the open simples, and the methanic zone, the closed simples, and there's no difference. Those 2 groups of organisms keep their substrates at limiting concentrations. Well, by the way, it's not the methanogens that eat these volatile fatty acids, that's the bacteria. The methanogens also don't touch the methane, they have bacteria to take care of that. But low micromolar range for the electron donor. So how about the electron acceptors, the sulfate? Well, if you do the experiment in a different way, I've here been starving the sediment for sulfate until all the sulfate was gone. And then you add a small pulse of sulfate, say 10 micromolar, the lower left, and measure then the sulfate concentration. It's done with radioactive sulfate over 10 hours. You can see the rate at which it disappears. And from that, you can draw another Michaelis-Menten curve and now it has an apparent Km value of 1.3 micromolar. So the apparent Km has decreased 1,000-fold by doing the experiment in a different way. So what is going on? Well, what is going on is that the top curves only show half of the story. They show what is happening at the high concentrations, but we don't have these rates down to very low concentration, so we don't see the high affinity uptake. This high affinity uptake could be due to other bacteria that start to grow as we starve the sediment of sulfate, or it could be the existing community of sulfate reducers that are able to shift their affinity depending on the concentration level. And that we have been able to show in a pure culture of sulfate reducers, a common marine sulfate reducer, the Sulfobacterium autotrophicum. This is a rather odd Michaelis-Menten plot because it's on a logarithmic concentration scale, but if you look at the sulfate reduction rate, you can see, let's just see I'll just put the pointer on. So this part you can see is the low affinity with a Km of 150 micromolar. Once the concentration drops to a few hundred micromolar, the bacteria turn on their high affinity sulfate transporters, and suddenly they have high affinity. The Km drops 20-fold, and this is part of what happens in such a sediment. And only when you get into the very low concentration range do you discover that. Is this important? Well, it is important for some things. If you look, for instance, at what we discovered was a cryptic sulfur cycle going on in the methanic zone below the sulfate zone. The question is, what is the sulfate concentration that is maintained there, where you have both slow sulfate production and oxidation? Well, we have done measurements of this sulfate in the Baltic Sea, where we have here the sediment core taken from an IODP expedition in 2013, going down to 100 meters on the scale. And you see the data points are sulfate concentrations measured in the methanic sediment. And we had another cruise where we took gravity cores and found out that the true concentration of sulfate there is 10 micromolar or less. So this shows here percent seawater concentration measured in the core. So what happens when your core is that you pull down also seawater, or drilling fluid is pumped down, and this diffuses into the core and contaminates all the pore water samples with seawater. This is what the microbiologists are very aware of, because that is also contamination of the microbiology, and they use a perfluorocarbon tracer to see what is the degree of contamination. And it's the same order of magnitude, especially in the periphery of the core. In the center, it's less, but that's because these samples are taken much faster than you can squeeze pore water out of the sediment. So this is contamination with seawater. And if we look at all the data in the database for sulfate in the methanic zone, this is the distribution. In gravity cores, it ranges from a few tens to a few hundred micromolar. In IODP cores, it ranges from a few hundred micromolar to a few millimolar. And no one has ever determined the true sulfate concentration in the methanic zone in IODP cores. So we cannot prove that this is contamination, but I'm very convinced that it is. I think the IODP has a problem with very general contamination, and it is important for some ions. For many ions, it's not really so important. So this was one discovery about the affinity of sulfate reducers for sulfate. Then how about temperature? There are many studies in the literature of how short-term incubation experiments of respiration, for instance with sulfate reduction, shows minimum, an optimum temperature of sulfate reduction, which is marked here with red. And then this sulfate reduction drops again. I can just mention this little blip here where it goes up again is because there are also thermophilic sulfate reducers in the Arctic sediments that start to germinate when you heat them and then grow. But I was always concerned that a temperature optimum at 25 degrees in a sediment that has never been above 2 degrees doesn't make sense. So what are we learning from these temperature curves by short-term incubations? We've studied that recently. And here's an example of 3 different fjord sediments from Svalbard and 1 from Northeast Greenland. And you see a similar curve here. The rate of sulfate reduction, the temperature scale goes from -5 to 35 degrees. And then the rate of sulfate reduction peaks in the range of 20 to 30 degrees, as we've seen before. But when you have a sediment that is more or less in steady state, it is not this short-term ability to have high respiration rate that is decisive for the competition, that it shows the adaptation. It's the growth rate rather. How fast do they grow? And we've been able to determine the growth rate in these sediments. And it turns out that the highest growth rate lies between 10 and 20 degrees. And if you look at this part of the curve, you can see that there's negative growth rate above 25 degrees. Above 25, they're dying, even though this is where you have optimum. So this optimum doesn't really show anything ecological about the organisms. They're desperately trying to respire before they die. But again, when you think about it under steady state conditions, it's also not really the growth rate, potential growth rate. It's the efficiency with which the organisms can use the substrate that they turn over to build new biomass and grow and outnumber the others. So it's really the growth yield that is decisive. And we have been able to determine the growth yield. And sure enough, the growth yield is highest between 2 and 10 degrees. Now finally it makes sense. These are truly cold adapted organisms. Their optimum respiration rate does not tell about their ecology. We need to do these more careful experiments, and we need to reconsider our interpretation of these many temperature curves that are in the literature at this point. So how do we determine the global rate of mineralization in the seabed, the global sulfate reduction? And the most recent budget that has been determined was by Marshall Bowles and co-workers in Science 2014, who found 23 teramole of organic carbon was mineralized anaerobically per year. They used sulfate data from the IODP database. They did not use gravity cores. They use a diffusion reaction model of sulfate profiles with a single exponential function that they could fit to the sulfate curves, which basically mean, to my understanding, that they selected concave down sulfate profiles for their modeling. And then they trained an artificial neural network using different relevant sediment and water column parameters, which was really a novel way of doing a global extrapolation and mapping. So let's see what you are learning from this approach. One thing we note is that the IODP does not drill on the continental shelf, but they have cores from all over the ocean. And here I plotted the frequency of gravity cores and IODP cores as a function of water depth. And you can see gravity cores are on the shelf upper slope, the IODP primarily in the open ocean, deep sea. And if you then look at the sulfate reduction or sulfate flux, and I've taken as an example the sulfate flux across 1 meter depth in the sediment, which is a measure of how much sulfate reduction is below 1 meter. You can see that it drops with depth in the ocean. That's the vertical scale down to 7,000 meters, and that's what you would expect. But if we plot this on a log scale, here comes the shock. there is not an overlap between IODP cores and gravity cores at the same ocean depths below 1,000 meters. There are no IODP data from the shelf, okay, but for the deeper ocean, the IODP cores show a sulfate reduction that is an order of magnitude lower than what you find in gravity cores. Now that was a surprise to me. So why is that? And one thing you notice then when you analyze the data is that the IODP cores are mostly from areas with a low sea surface chlorophyll, while the IODP cores, the gravity cores are from much higher sea surface chlorophyll. And when you look at the sedimentation rates, the discrepancy is even much larger. IODP cores are from regions with low sedimentation rate, gravity cores from regions with high sedimentation rate. Did you know that? I didn't. I'm wondering why is that? Of course, each expedition has a special motivation for why they choose their site. And in general, I think IODP expeditions choose sites where you can study paleoceanography, plate tectonics, things where you need a deep time window in order to get a long geological record. Geochemists who take gravity cores maybe more go to the hotspots where you have high diagenetic rates and interesting processes. But it means that if we choose only one type of cores, we get one picture of what sulfate reduction is in the ocean. So then the next question is, if we then only use the concave down sulfate profiles for the modeling, then are we excluding anything by that? And whoa, I have here normalized the entire database of complete sulfate profiles to 100% sulfate at the sediment surface and 100% depth where sulfate ends. So this is the entire sulfate zone. And it's just to show the shape of the sulfate curves. And what it shows is that half of all the curves are concave up. They should be concave down in order to model them. And when you start to look in the literature, you find out that people are not using these concave up curves for modeling. They pick the ones that you can really model. And no one wants to model that there is sulfate production in the seabed. So there's a real bias in this. And I don't think anyone has ever tried to compile this before. I was shocked. So I think we really need to go back and look at how we select curves and why is there such a high frequency. I know in some cases there are good reasons why they look like this. Here, for instance, are some cases where there have been mudslides or turbidites that have added sulfate-rich sediment to a thick layer, but then you would think this is something special for the upper slope. It could also be bioirrigation, but I've tried here to split up the data to different water depths. 0 to 50 meters, the upper two are from the continental shelf, the others from the deep sea. It's the same everywhere. I've tried to split it up according to the depth of the sulfate zone. It's the same everywhere. It's not related to bioirrigation or to mudslides or such. It is universal that half of all the sulfate curves are concave up. Why is that? And here's the same with the IODP cores. They don't core on the continental shelf, so the top is empty, but for the deep sea, it's the same. So I think we have a real piece of homework to do to find out why is this. So I hope some of you young people are listening, because I'm really curious for someone to start looking at that. So if we then want to understand the global rate of sulfate reduction, we need, as I said, to distinguish between gross rates and net rates. Gross rates are what is mineralizing organic carbon. Net rates is the difference between gross rates, and then the reoxidation of sulfide. And the number here, global sulfate reduction, is taken from Don Canfield, who estimated 45 teramole of sulfate being reduced annually in the global seabed without these vegetated coastal areas. And if we look at the distribution in the ocean, the continental shelves are really counting heavily. 60% of the total global oxygen uptake of the seabed is on the shelf. 66% of the sulfate reduction on the continental shelf. It's 77% if you include also the vegetated coastal zones, and 75% of the methanogenesis on the continental shelf. So with these numbers, I've tried then to compile a global budget here. And these are numbers I picked partly for total net primary productivity, 4,300 teramole organic carbon produced in the upper water column, the biological pump, the settling of particles out of the photic zone, 500 teramole organic carbon, but then our own number for sediment oxygen uptake that we have estimated recently, 212 teramole organic carbon mineralized annually in the global seabed. Then there's this estimate of sulfate reduction that I just showed you that translates to mineralization of 77 teramole of organic carbon per year. And that means that about 1/3 of the mineralization of organic matter in the seabed is through sulfate reduction. So that's the estimate. The estimate by Marshall Bowles that I mentioned is 23 teramole. And this was by modeling, so that's a net rate that they estimated. What is the sediment burial? Well, I picked one number from Klaus Wallmann and co-workers, which is 12 teramole organic carbon. There are other estimates that are somewhat higher, but this means that of all the organic matter settled on the seabed, about 5% is buried over Holocene time. Methane production, well, the methane flux globally is 5 teramole organic carbon per year. And you can see that that corresponds to about 7% of the global sulfate reduction. So 93% of the global sulfate reduction is organoclastic, and only a small fraction due to methane. Then Antipouitos and Frank Wenthoff have estimated up to 1.7 teramole organic carbon coming out with the methane cold seeps. And then again, Wallmann et al. An estimate of the burial rate of methane hydrate, 0.5 teramole, meaning that 10%, or it is less, 7-8% of the total methane production in the seabed gets tied up in gas hydrates. The rest is either being oxidized in the sediment or comes out of the sediment. And then finally, burial of sulfur, reduced sulfur in the seabed, 2 teramole of organic carbon is the equivalent of that. And you can see that this is about 3% of the total sulfate reduction of the 77 teramole. 3% of the total sulfate reduction is tied up as reduced sulfur in the seabed. That means that 97% of all the sulfide produced by sulfate reduction is reoxidized back to sulfate. The sulfur cycle of the seabed is almost completely open and, well, closed, you could say. And this, I think, indicates why is there such a great difference between gross and net sulfate reduction. And with that, I just want to end up with thanking all the people who have contributed to these studies. I have indicated all the publications from which I have taken this information. It is all published. Well, some is just submitted. With that, I thank you for listening.
Thank you, Bo. Great lecture and congrats on the medal. We have time for a few questions. Yes, can you please go to the microphone? Oh, sorry.
For your presentation. I have a question, but you highlight that there is a very important cryptic sulfur-cycling, so it's basically sulfide and back to sulfate. But sometimes it seems that this cryptic cycling occurs in the presence of iron 2, we saw iron 2, but the iron 2 is supposed to react with sulfur very rapidly because of oxidants. So I was wondering, how do you explain that the consortium of bacteria are still able to reoxidize the sulfur and replace to the kinetics of McNamara or iron sulfur classification?
Yeah, a very good question, and I wonder by that myself. I have, when we first published the paper on the cryptic sulfur cycling, we could not explain how could there be such an efficient reoxidation of sulfide down in the reduced sediment. It's, I think, still an open question, and it is also argued that there's not enough burial of iron 3 to account for this, but I showed you the power law. Hardly anything is happening down there. It takes so little iron 3 that you cannot see the change in concentration from that. But this is really an open question. Someone needs to go in now and understand how does this happen. I don't understand it. Yeah.
I'm intrigued by your evidence of high affinity and lower affinity sulfate reduction depending on concentration. And I wonder how this fits with evidence from isotope fractionation and sulfur isotope fractionation. Basically, we look at it. Latin kinetic is the constant is when the capture is equally fast as the release. And conceptually, so that means if there are high affinity transporters that capture the sulfate and process it to its sulfate reduction. Now, isotope fractionation tells the story of how much of the whole chain is reversible. The higher fractionation, the more slots are reversible. At some point, more AT exchanges and feeds back into sulfate. Is there already some data to link these two lines of evidence and to see how much of the sulfate reduction would be reversible or how much of the chain would be reversible for the high affinity situation at low concentrations?
Yes, well, there are different aspects in one question here, and one question is what is the cell-specific rate of sulfate reduction, right? And that so that is one part of the regulation. The substrate concentrations, well, often mostly high sulfate, so that doesn't affect the result, but very low substrate production rate. And that is what controls the cell-specific rate of sulfate reduction. The substrate concentrations do not vary much, so that's not the secret here. So it's the rate of the production of the substrates that is decisive. And there are many experiments, I know, showing low cell-specific rates, higher fractionation. That makes sense. But I don't think we understand really what controls these low rates, other than the overall rate of their electron donor production.
It's also a question of energy balance. So each transport of sulfate into the cell costs energy.
Yeah.
So each sulfate molecule lost out of the cell is a loss of energy.
Yeah.
So the more reversible the whole process is, the more energy is lost in the whole process. I wonder whether that's linked to this high affinity transporter and how this translates into propagation towards sulfide, or whether it's a leakage to the what extent as
Yes, but the high affinity sulfate transporters kick in only when you have extremely low sulfate concentration. And in all these studies of isotope fractionation, you are not working in that concentration range. So it's a different story. Yeah.
Thank you for the fantastic lecture. There's another curious side about sulfur-reducer, and despite this thinking they control so much of the sulfur in the seafloor, they are so bad in degrading organic matter of all kinds. They just potentially, and I haven't followed the recent literature, but they can do it. They can never act alone, except a lot of other microbes prepare food for them.
Yeah.
Is there any move or any advances on that part? Why you can be so efficient? Why so relevant to our cycle? But you just depend on everyone else to provide a bit of formal acetate hydrogen, which is of course another missing link in knowing what is going on.
They get a little help from their friends. Apparently, there's not a great evolutionary pressure on having a very diverse ability to use different a range of substrates. So these are syntrophic relationships that work perfectly well. It's even much more extreme with methanogens. Acetate is the main substrate for methanogenesis in marine sediments, but they don't touch the methane, the acetate. They have bacteria that take care of that and serve the electrons for them. Syntrophy is a wonderful concept in that respect. They don't have to be universalists. Mm-hmm.
We're running a bit late, but I would like to end with the online questions. Are the sulfate profile that are not concave associated to high sedimentation rates? That's an online question. So are the sulfate profile that are not concave associated with high sedimentation rates?
No, not at all.
No, not at all. Okay.
There's no relationship.
Okay.
That I have been able to find so far.
Okay.
Yeah.
Good. Thank you so much for the great lecture again. So our next speaker.